Hi, my name is Caitlin Britell, and I will be discussing GABA-enriched tomatoes. This is a research project for the Animal Embryology and Biotechnology course at the University of Connecticut. GABA-enriched tomatoes are also known as the Sicilian Rouge high GABA tomato. They are genetically edited to contain increased amounts of the gamma aminobutyric acid compared to the average tomato. This is done using CRISPR-Cas9 technology and is the first food edited with this technology to be put on the market. These tomatoes were created by the Sanitec company, who received approval for their product in 2020 before releasing it to the public in Japan in September 2021. These tomatoes were made because of how common GABA-enriched foods and supplements are in Japan. Over 400 food and drink products are currently being sold there. GABA is an amino acid that acts as an impulse-blocking neurotransmitter. The factor that drives many to purchase these products is the possible benefits related to overall relaxation. There are claims it can decrease blood pressure, improve sleep quality, and decrease stress and anxiety. This variety of tomato is created using CRISPR-Cas9 gene editing technology. Guide RNAs are specifically designed to target sequences in the CGAD2 and CGAD3 genes, two of the genes that code for glutamate decarboxylase, or GAD. This enzyme plays a role in GABA synthesis. The Cas9 enzyme creates a double-stranded break in the DNA, and stop codons are introduced in breaks upstream the autoinhibitory domain, causing a deletion of the domain sequence. This area of the gene is linked to the inhibition of GABA accumulation. Initial studies done by Nonoka et al. In 2017 found a 7 to 15 times increase in GABA accumulation. Smaller mutant lines were also found to have high GABA levels in their leaves, which aligns with previous studies done with GABA accumulations in plants. The graph represents the GABA levels of 3 different mutations, each of which had 8 different mutant lines. Varying levels of the amino acid can be observed in each line, and most lines have elevated amounts compared to the control group. A handful of problems arise when working with GABA-enriched tomatoes. Based on the current studies available, there is a possibility that the accumulation of this amino acid in plant leaves can have size and yield reducing effects. There are also suspected impacts on cell elongation, which could play a role in size and yield. There is also a lack of regulatory policies surrounding gene editing food, making it more difficult to commercialize in countries outside of Japan. Improvements in this gene-edited product have also been proposed. The use of tissue-specific promoters can allow metabolic pathway activation in fruit tissue only, so there is no accumulation in the leaf tissue. Base editing could also be a more accurate way to edit GABA-related genes without seeing any undesired effects. Increased GABA levels may also trigger pathways in plants that improve resistance to certain environmental stressors. Previous methods used for increasing the GABA level in plants include traditional breeding, fermentation, and genetic modification, or GMOs. Traditional breeding is very difficult to work with, as it takes longer periods of time and results cannot be guaranteed. In fermentation, microbes such as lactic acid bacteria can increase GABA levels, but it is not to the same extent as other methods. GMOs have also been utilized, but this method is less controlled and subject to stricter regulations. Overall, producing GABA-enriched products from CRISPR-Cas9 seems to provide the most efficient and reliable results. The pending release of these tomatoes was important to many, as it encouraged Japan to create regulations surrounding genome-edited plants and foods. This opens the door for the approval of other genome-edited foods and encourages other countries to create their own regulations. 2 CRISPR-Cas9 edited fish have already been approved in Japan since 2021. This product also makes GABA supplementation more accessible to the public and reduces the need for synthetic supplements. The stress-resistant effects of increased GABA levels in plants also make this crop more sustainable than the average tomato. The increased resistance of these tomatoes improves yield and results in a reduced burden on resources. Overall, GABA-enriched tomatoes and other CRISPR-Cas9 edited foods could have a positive impact on consumers and agriculture. Thank you for your time.